One very popular method for analyzing protein sequences is look at the distribution of some properties that can be given numerical values. And one way of doing that is try to count a kind of a running average, or use a window over which the average is calculated, in the same way as in listing COVID cases. There's no sense in listing the daily averages, which are jumping back and forth, and there might be a problem in the statistics of the weekend, so that the Mondays always show a triple amount, etc. So instead, they are giving a windowed average of 14 days to give a smoother curve to show the real trend. And in the same way, if we look the things like hydrophobicity, the non-polarity of amino acids, they are going back and forth. Usually it's common to see that every second amino acid might be hydrophilic, every second hydrophilic, but for a better trend of a hydrophobicity profile, you take a window of say 11 amino acids and calculate the average of that. And you can use the same approach for any property, as long as it's something you can present as a numeric value, so that each amino acid type has its own value given. There are similar but slightly different scales for hydrophobicity. Some of them are called surface probability, and there are many different versions of the actual hydrophobicity numbers. But the idea in these profiles is that when you are looking for a feature which is X amino acids long, so it's a good idea to use a window which is half of that number in length. So for detecting transmembrane helices, which are continuous hydrophobic sequences of amino acids of about 20 in number, or maybe 22, 23, depending on the membrane thickness. So then a window size of 9 or 11 residues might be good. And here's one example of a rat olfactory receptor. And now this is supposedly a 7 transmembrane helix receptor, but how many helices do we see? So let's try to see the peaks. 1, 2, 3, 4. Is this a peak? Maybe not. 5, 6, 7, but we are not quite sure. Maybe here before number 100 we have something, and maybe this is a peak as well. So this is not all so certain. And we have a tool in UniProt, which is, or actually Expasy connected to UniProt, which allows us to try that. And we have a nice demo of that in a recording, so I'll skip the demo part. But in that, I recommend that you try it on your own. You'll see how the changes in the window length and other parameters might change the aspect of the plot. But still, this is not quite satisfactory. The reason for that is that when you have a single transmembrane helix, it's very clean. It's leaning on the membrane lipids from all sides, and therefore it has to be hydrophobic all through the 20 amino acids. But for a bundle of 7 transmembrane helices, we have the helices which are touching each other, so that we have some sides of the helices which can have some ionic contacts or hydrogen bonds, so that we have some interruptions in the pattern of hydrophobicity, and therefore the numerical approach is not that good. But instead, when we do some machine learning for what kinds of patterns we see, so the 7 transmembrane helix pattern is very easily learned and very easily recognized in all of those 7 helices, despite the the exceptions in hydrophobicity. And same holds true for other families of membrane proteins. And we have two very good programs which are required to be used in, actually just the latter one, TMHMM, is required to be used in homework. But for curiosity, the other top-ranking program, HMM Top, is of Hungarian make in one of the universities of Budapest. But both of these programs are technically using hidden Markov models in which they have a system of states which are assigned to the amino acids. So there are membrane and non-membrane states. Maybe there are even states of transition on the membrane surface going from inside to the outside of the membrane. And a hidden Markov model algorithm can test the sequence to find the most probable sequence of states which matches the sequence of alphabet characters. It sounds complicated, but still this is a method that works pretty nicely, and there are cool algorithms. One of them is called Viterbi algorithm, which tries to align your actual sequence to the hidden sequence. Well, no, you don't know the hidden sequence, but it's trying to discover the sequence of states, and the output is the most probable states. And if you do a filtering so that you try to find a sequence of 20 characters which have a continuous intramembrane state, so you have discovered your transmembrane segment. Well, you are okay if you forget all about this after this, but the important thing is that this is one of the best prediction tools for getting some biological and biophysical features from protein sequence alone. So these have more or less 90% success rate in the prediction of transmembrane helices in eukaryotic cells. And the output of TMHMM looks like this, and this is a very neat, clean pattern showing just a single transmembrane helix. I think this was for carbonic anhydrase 12. There's a tiny intracellular domain and a large extracellular domain, which includes the catalytic part. And interestingly, we also have a prediction for outside and inside probability, and we can see that there's a correct prediction of having all the N-terminal part to be outside, and the C-terminal end is in the inside. And let's see if we can do the TMHMM prediction with the same rat olfactory receptor from which we got these quite dirty examples of transmembrane helices with the simple windowed calculation. So let's do the demo for predicting the transmembrane helices of the rat olfactory receptor. Here is the UniProt page, and if we look at family and domains, we may have, sorry, it was not here. We have the topology and domains under subcellular localization, so you could cheat and find the locations of the transmembrane helices automatic annotation, which has been done, inferred from a sequence model. It doesn't say what algorithm it has taken, but we just want the sequence. I click the sequence on the left and going for download of the sequence. This is the sequence in FASTA format, and just copying from here. And then I have opened the pages of the latest version of TMHMM, which is called Deep TMHMM, indicating that it has used some deep learning methods for training the neural networks. And just copying the sequence and pressing run. It takes a short while, but not too long. This is something which is free for academic use, so if you're really interested to analyze a large number of sequences, you may want to install the program in your local computer to do the work. But for a few dozen sequences, it's no problem to do the work in your own terminal. Do it in your web browser. And the result is here. And we have the most likely topology here with nice squared columns indicate that decisions have been made. So if you compare it to the posterior probabilities, which are the actual calculated results, you can see that there is some narrowing at the top and some broadening in the bottom, but they've taken, I guess, more or less the half height width to represent the most likely topology. And we also have the predictions for inside and outside probability, and these are again very logical in that we have every second region between the transmembrane helices being in the inside and every second being in the outside. And this is one of the weaker parts of most prediction tools, that the topology might be inverted, so that instead of having the as here outside, inside, outside, it could start by inside, outside, inside. But anyway, this is the result, and this should correspond to what we see in the UniProt, and this is way cleaner than the result we had in the not this one, the one which comes from the simple hydrophobicity analysis using the Kite-Doolittle scale for hydrophobicity. Hydrophobic is up, but this is kind of similar. But still, this is way more certain. We don't have to be second guessing if a kind of a narrow peak would actually be a real signal or not, or whether a split peak, which shows as two different ones, is two peaks or one or no peak at all. I think the region which was just before amino acid 100, this is the one that had the split peak. And then there were some very broad peaks, and this gives us kind of reasonable cuts which match the typical lengths of the transmembrane helices. And there it is. We have the result in many different versions as a one-line representation of the whole sequence, or as a table showing the helices and the inside and outside parts of the sequence. Okay, let's move on. We can go to the topic of protein motifs. This is like a part which was completely missing from the previous recordings. So when we talk about protein sequences, when we analyze the conservation of a number of related sequences or a full protein family of maybe dozens or hundreds of sequences, there seem to be some kinds of patterns or motifs in the sequence which are typical for that protein family. For example, in carbonic anhydrases, one of the active site motifs is histidine, something else histidine, which can be represented as HXH. And there are many ways of representing such motifs. We can do literal representations like concrete sequences, like NFS would be one instance of an N-glycosylation site. But if we want to give a more generic presentation for all possible glycosylation sites, it seems that the asparagine is needed. Asparagine is the amino acid that receives the large oligosaccharides in so-called N-glycosylation. And then we have any amino acid, but not proline, and as the second amino acid after the asparagine, there is serine or threonine. And we could represent this in a Unix style regular expression showing that it's asparagine, not P, and either of S or T. Then we could also have a presentation like ILV in square brackets, meaning any of these three residues, and then curly brackets that indicate how many times the amino acid is repeated. So this would mean 2 to 4 times of either of these medium-sized hydrophobic amino acids. And this is cool for short sites, and this has been an approach used in a ProSite database that was one of the first and quite successful. But this is a pain when we go to longer motifs. We might need more and more exceptions. When new representatives come up, you need to make the protein a little bit broader so that it accepts all the new members, and still not too broad that it starts finding the pattern in unrelated sequences. So instead of trying to find a generic way of representing the pattern, so you could just simply collect all the instances of the pattern and use enumeration of the patterns, the whole collection of these patterns. But often it's done in a more compact way so that these sequences are given as a table across the pattern, and then you calculate the percentage of each amino acid in each column, and this gives us a frequency profile, amino acid frequency profile for that particular collection of sequences. And there are mathematics for aligning a sequence to a kind of a frequency profile. And also you can use more developed statistical methods like hidden Markov models, which we discussed with transmembrane proteins. We can also construct a hidden Markov model for any kind of sequence motif or a complete protein domain, and then the Viterbi algorithm can help us decide if a new sequence is a new member in the family, whether it is statistically significantly similar to this hidden Markov model so that it would match. But anyway, the take-home message is that there are many methods of doing this protein motif analysis, and there are many motif databases that have been developed using different approaches or mixtures of approaches. And the most important one, which I'd like you to remember, is Interpro, which is a collection of databases all made for the same purposes, and having a kind of a consensus jury voting for you, giving different methods to analyze your sequence gives you more confidence to identify what your unknown sequence might represent. If we have a new genome which has been sequenced, or maybe a transcriptome that has been sequenced by RNA-seq, we'll talk about this in the genomics course. So we have a bunch of protein sequences that need to be annotated, and instead of having the bioinformatician to run blast searches one by one, it is much easier if we have a program which will analyze each protein against Interpro, and then we will have probabilities for different types of protein domains, and also by extension their associated functions with statistical significance built into the search. And so if your unknown sequence picks a hit for alpha carbonic anhydrase of the eukaryotic type, and your sequence comes from a eukaryote, then you can be fairly confident that, okay, this protein is an alpha carbonic anhydrase. There might be, if your organism is a vertebrate, there are profiles which are narrower, so that it will tell you if it's a part of the subfamily of CAs 6, 9, 12, and 14, or whether it's a CA5-like sequence, etc. And nowadays we have the conserved domain database developed at NCBI, also included in Interpro. But the CDD has its independent existence and development, and it is something we see in the context of BLAST searches. I'm not sure if you saw that when you run BLAST, a kind of a graphical summary which shows you domain hits in addition to hits to your query sequence. Okay, let's do a short Interpro search, and let's see what kinds of motifs we might find in a protein sequence.
I'm sorry, I have a question.
Yes, please.
About motifs, and the question is if they are like the transcription factors when they have the ting finger and helix turn helix. These are the motifs, right?
Yeah, helix loop helix,
Yep.
or yeah. So yeah, these would be other examples of the motifs which also have a structural interpretation, but you can recognize these motifs also on the sequence level. They have some residues which are conserved, or physical properties which are conserved, so that you know that certain positions need to have hydrophobic residues to make hydrophobic contacts, and then the zinc binding residues are only probably of acidic and amide type amino acids: aspartic acid, glutamic acid, asparagine, glutamine. So these are easy to see by sequence analysis. Once you've analyzed a number of known cases, you can generalize it into a hidden Markov model, and you don't have to do it because people already did that in one of the member databases in Interpro. But let's move to Interpro. I'm going for screen sharing, and this is Interpro. And you can do many things in Interpro. You could just start by a text search to see what kind of domains are known for zinc fingers, for instance. Search by text. And let's see what we find for zinc finger. Okay, 3,000 entries which mention zinc fingers. They have the zinc fingers are kind of common, so that they are found in many different proteins. You can see that we have different proteins which contain the zinc finger motifs, and these are domain signatures which identify the entire domain for whichever of them. So this is kind of complicated, and maybe the zinc fingers as such are so short that it would be hard to get statistically significant results, but we can see clearly quite a few results here. And here we can see that these signatures come from many different originating databases. Some of them are from the original Interpro, and then the larger Interpro also includes PFAM and Panther and data and signatures collected from the protein information research protein information resource that used to be an American protein database, but it merged with UniProt about 20 years ago. But there's a lot of things we can find. Let's go back to the sequence search. So this is the main page of Interpro, and let's try what kind of motifs we find in the rat olfactory receptor. And let's go for just search with the default options. Okay, it is still searching, so we don't have the results yet.
I have another question. I'm sorry.
Yes, please. Yeah, well, it's good to talk while the searching is going on.
Okay.
Yeah.
So as much as I remember, domains and motifs are not the same, but they both are like a super secondary level of the protein or something like that, but they are not the same, right?
Well, again, there are different levels of studying. So if you are looking at structural motifs, so a zinc finger or a helix loop, helix would be a structural motif, and these would be super secondary structures, combinations of short secondary structure elements which are still not like complete domains, which would be at the level of tertiary structure. But when we talk about sequence motifs, so the sequence motifs can be fairly short, so they can only recognize maybe a single phosphorylation site or a short active site in a protein, or the N-glycosylation site, etc. Or they could be long motifs which recognize a whole domain. And the most useful ones for classifying proteins and doing functional predictions for new proteins, the most useful ones are the databases and patterns which contain statistical models for complete domains. So they are kind of overlapping. So a domain signature is just a longer sequence motif.
Okay, I see.
Which is represented most of the time with some more complex data structure like frequency matrix or a hidden Markov model. Let's see if our search results are there. Okay, done in less than 2 minutes. Now, where are the results? Here. Okay, as one thing, we do have predictions of the transmembrane helices, or rather, oh, this is annoying. I'm mouse overing all the time. Oh, come on. Leave my sequence alone. But the top lines are showing you 7 matches which are to G protein coupled receptor of rhodopsin type, so that is the 7th transmembrane helix receptor prototype, bacterial rhodopsin and rhodopsin of the visual system, and this domain has been described by the transmembrane helices, and no attention has been paid to the parts which are in between the helices. So the matches are discontinuous, but still they give you membership in this family. And then we have others, like the green one here is olfactory receptor. Okay, so that's good. We know that it's related to the sense of smell. And here's another one, which is a continuous domain for G protein coupled receptors and G protein receptor family 1, and I wonder if 2 is a subfamily. Family A, G protein coupled receptor like. So there are many ways of analyzing these proteins or classifying into families, and we get the many prediction results like the transmembrane domains and cytoplasmic and intracellular domains, etc. So this is giving some of the same information we already have in UniProt. At one point, TMHMM was the tool that was used for transmembrane predictions, but I guess that for these olfactory receptors, they are using a specific 7 transmembrane helix receptor family transmembrane domain pattern to get the exact positions. But there are many different finds here, and more or less all of them tell you the same thing, that this is obviously a 7th transmembrane helix receptor, and some of them are more specific, discussing a particular subfamily. One of these is telling that this is an olfactory receptor. Let's click on this one and let's see where we go. No, this was just a link to show where in the sequence we had the domain hit, which we already had in the graphical form. But what I wanted to see was the actual interpro. Okay, the links are on the left. This is the olfactory receptor, the green one, and clicking on the IPR link. There we go. This shows the description of the whole entry, and it shows what is the kind of larger family where this belongs. This is the rhodopsin-like receptors. And then we have some text which helps you understand your sequence even more. And having found this domain, the protein is very likely to be included in specific biological processes and molecular functions. And the cellular component, being a membrane protein, is also made obvious from the result. So this helps you understand how the databases are built and how sequences are annotated now that you've seen INTEPRO in action. And so the homework you have for the topic of sequence analysis is presented in a PDF. You have a data file which you need for doing the work, and we have the submission box here. And the submission is recommended to be by next week, but anyway, it's a soft deadline, so don't worry if you don't make it exactly. And this is the PDF. And we have all the links for predicting signal peptides and different peptides for targeting to mitochondria or chloroplasts, except these are human proteins, so you can disregard chloroplasts. We'll talk about what are GPI anchors in a minute, and transmembrane helices we've covered with different analysis tools. And the challenging part here is that once you have your predictions of GPI anchoring, signal peptide predictions targeting to mitochondria and transmembrane helices, so you have to kind of figure out on your own how these different positive results of the predictions fit together and what would be the actual targeted localization of each of the CAs 1 to 14, which we have there. So you are asked to give your final analysis of each of these 15, the CA5 with variants A and B, or two different genes A and B, and also report a little bit, a few lines of text explaining how you could come up with your conclusions. And there are some questions. Some of them might be a little bit complex, but if you are not able to find answers to them, so feel free to ask in the forum after a couple of Google searches that didn't take you anywhere, or consulting a cell biology textbook if you have one. And then we have some questions regarding INTEPRO, and there's a tool for different kinds of protein predictions. And this is annoying in that they only give you 3 free trials in some time, maybe a month. So maybe it would be a good idea to save at least one for your project and just do one test with one of the CAs. And here's a recommendation of not choosing these 5, which are kind of too easy cases. So you would rather go for one of the others. And also we have the site of prediction services at the Danish Technical University, which has now some integrated tools for getting the localization for all features predicted based on the presence of all features. So you might want to use that one as well. But now let's talk about the subcellular localization and how they come about biologically. If you have studied as a cell biology major, so this is something that will be very familiar for you, but if you are a pure biochemist or whatever else, so this might be useful for understanding the sequence analysis homework. Well, one thing is that eukaryotic cells are complex. There are many different compartments, and there's an organized system of transporting proteins from the site of synthesis, usually the cytoplasm, into the different compartments. And there are some proteins which are resident in a compartment, and there are some compartments which are linked to transporting proteins, like the secretory pathway. It has its machinery which stay there, and a lot of proteins which are passing through in order to come out of the cell or to go on the cell membrane, on the cell surface. So there's an active system of sorting and transporting proteins, and many of these systems recognize parts of the protein which can be also recognized by sequence analysis, and that's why bioinformatics is kind of successful in getting the idea of where in the cell a protein might be present, even in the absence of experiments. Obviously, experiments have been needed to identify the proteins by immunology and microscopy and so on to get some examples of true positives. And once we have them, so we can analyze what are the common features of the proteins we have in one place, and in that way we get the protein motifs that are linked to specific compartments. And some of the possible compartments are shown here. This is more or less high school biology as well. So we have the cytoplasm, which is like the default location where proteins end up if they have no sorting signals. They just stay in the cytoplasm. Protein synthesis takes place in the cytoplasm, and the ribosomes leave the protein there, and unless something happens. And let's talk about some of the possible fates there. Well, the secretory pathway is interesting in that it involves a signal peptide, which is about 20 amino acids in the beginning of the protein in the N-terminus. And the secreted proteins are not synthesized to be ready-made and folded in the cytoplasm, but instead, as soon as the signal peptide is coming out of the ribosome, we have proteins binding to the signal peptide which arrest the translation. And once the ribosomes with arrested translation are floating to the endoplasmic reticulum, or maybe there is even some kind of an active transport guided by the signal recognition particle. So the signal recognition particle helps the ribosome dock on the endoplasmic reticulum. Let's go back and show you that ER is the kind of network of membranes which is all over the cytoplasm. We have smooth endoplasmic reticulum, and then we have rough endoplasmic reticulum, which is like the continuation of the nuclear membrane. So this is rough endoplasmic reticulum, and it is rough in here is the term. It is rough because it's decorated by ribosomes which are attached on the surface of this ER, and they are finishing the translation on the surface of that membrane. And the inside of the ER is like a different world compared to the cytoplasm. There's a different pH, different set of proteins, and there is an intact membrane enclosing the inside or the lumen of the ER. And once the protein is in the ER, it doesn't normally come out of the ER. Instead, it's going forward into the secretory pathway. So we have the translation stopping, and then the protein is then translated all the way to the end while it's on the surface and of the ER, and the peptide is passing through the membrane in an unfolded form, and it's only folded in the inside. And after the whole protein is synthesized, it's folded and it receives its final form. And even before it's folded, we have processing happening. The signal peptides are cut off as soon as the protein enters. We have end glycosylation happening on the unfolded protein, etc. But anyway, then there is a trafficking pathway going from ER to the Golgi apparatus. It's here, it's disconnected from the ER, and it means that proteins have to be packed into kind of membrane vesicles, enclosed small balls or spheres with protein content. The vesicles are fused to the Golgi, and then processing goes on inside the Golgi, and we have the modifications of the sugar chains of glycoproteins happening there after the initial end glycosylation in the ER. But this is my favorite topic, and I could go on like a course of several weeks on this alone, so I need to stop here not to go to too many details. But anyway, the presence of the signal peptide is something that we can recognize on a sequence level and identify secretory proteins, and then further sequence motives can help us to recognize the proteins that stay in the plasma membrane. They need to have the transmembrane helices there, and other motives are available for recognizing proteins which are residents of the endoplasmic reticulum or Golgi. The movement to lysosomes also happens in the secretory pathway. So from the Golgi we have transport into lysosomes, but again, too much details. And then we have the targets of like mitochondria and chloroplasts, the nucleus, and there's also some proteins which are on the internal leaflet of the plasma membrane. But we are not studying the nuclear transport. These signals are kind of weak and not so easy to get without errors. So because the tools are not there, we are not going to try it. But if you get the nuclear localization using the tools at the DTU, so feel free to report that as well. But we would be just classifying cytoplasmic mitochondrial transmembrane and secreted in the data set we have for the homework. Okay, regarding membrane association, the transmembrane helices are the most common and a very typical way of having the protein to stay connected to the membrane. This is so hydrophobic, so non-polar that it doesn't fit in a water environment thermodynamically, so it's just stuck with the lipids and the fatty acid change in the inside of the lipid bilayer. We have some barrel-like proteins which are composed of beta strands that have a hydrophobic outside and a hydrophilic inside, and these are often pores which allow things to pass through large ones by passive diffusions, and these are stuff you find in bacteria. The proteins are called porins because they make pores or tiny holes in the membrane. Some proteins can be associated to just a monolayer so that we have a kind of amphipathic alpha helix, which is like a log with one side liking the water and the other side liking the air, or in this case it is one side liking the oily lipid inside of the membrane and the other one is the aqueous environment outside of the membrane. Then we have lipid-linked things in the cytoplasm and in the outside, and this GPI anchor is one example of these lipid-linked things. And then we have peripheral membrane proteins which are attached to another protein which is an integral part of the membrane. But these are very hard to predict. You would need to look at the interaction databases or read the UniProt entry to tell you that a specific protein is part of a protein complex which is associated with membranes, like the chloroplast photosystems, which are of dozens of proteins, some of which are attached to the membrane and some of which are attached to other proteins.
I'm sorry, can I have a question?
Sure.
RG coupled receptors are of this kind, so this protein attached, do they classify as that?
Yes, there would be transmembrane proteins, and with 7 of these transmembrane segments instead of the 3 shown in this example.
Okay.
And instead of having the segments going like on the side of each other, instead of them being like, instead of them being a kind of a plate, they would form a bundle, which is like one helix which is in the middle and 6 which are around it. But this is a minor detail. But anyway, they are transmembrane proteins, and that's why TM-HMM is very good in finding the segments which are assumed to be within the lipid bilayer. Okay, GPI linkage, that's short for glycosyl phosphatidyl inositol anchoring. And the phosphatidyl inositol is a phospholipid which has the fatty acids. There is a glycerol here, and there is a phosphate, and now instead of, as the hydrophilic head group, it has a sugar-looking thing, inositol, but actually this is a cyclic alcohol with 6 OH groups. And this GPI can take additional sugars here, which are then part of the GPI anchor. And okay, we only have the schematic for the GPI anchor here. And you don't need to worry about the molecular details. But the important thing is that this glypiation, as they call it, is initiated by a signal in the C-terminus of the protein. It's another hydrophobic segment which is then cut off inside the ER, and it is exchanged for this GPI anchor. And most of these GPI anchored proteins are soluble. I think all of them are. They are soluble before the glycation happens, but once this has been added there, so the protein is locked to the membrane. This lipid is part of the bilayer, the outer leaflet, and it's holding the protein anchored on the membrane. And there's a little bit of a linker there, so it's not right next to the membrane surface, but there is some room for moving the protein. And again, the sequence features of this GPI anchoring recognition sequence, they are recognizable, and we have a website from the University of Bologna which has a fairly easy tool for finding the GPI anchored sequences out of your data set, if any. So this is the case C here. And because of the vesicle traffic, what is in the inside of the endoplasmic reticulum, it is like one membrane across from the cytoplasm. When the vesicles are splitting from the ER and Golgi, they go to the plasma membrane and they fuse there. And once the fusion happens, the inside of the ER becomes the outside of the cell. And so whatever proteins were soluble inside these secretory vesicles are released to be free in the outside world and to do some work as signaling or whatever, or maybe building the extracellular matrix. And in case of membrane attached proteins, they remain on the membrane. But the proteins which are in the GPI linkage are always on the outer surface, the outer leaflet of the plasma membrane. And as a curiosity, because they are with very long fatty acid chains, they are enriched in the so-called lipid rafts, which are also enriched in cholesterol and glycolipids. But again, that's another detail you have no practical use for. But you might impress somebody in a bar by showing some trivia about cell biology and membrane microdomains. But the important part for our course is recognizing the GPI linkage. In addition to the sorting signals, we discussed transmembrane helices, signals for mitochondrial localization, mitochondrial targeting peptides, and the GPI anchoring recognition peptides. So we have some others. For instance, the resident peptides of endoplasmic reticulum curiously have a 4 amino acid literal sequence: lysine, aspartic acid, glutamic acid, leucine, which would be KDEL in the one-letter notation. And for lysosomes, some proteins have protein signals which are again kind of vague, like the nuclear transport signals, but some carry a sugar tag, which is a specific phosphorylation of mannose, one of the monosaccharides in the glycoproteins. And this mannose 6 phosphorylation happens in the endoplasmic reticulum. It goes to one specific mannose in the glycoproteins, and once it's there, it's like a barcode for the sorting machinery, so that the vesicles going to the lysosomes will be enriched with the proteins with the mannose 6-phosphate, and they will be taken there using this barcode or address label as the targeting principle. But this is all I wanted to say of the kind of biological, cell biological and biochemical background for subcellular localization, and I hope referring to the content of these slides and finding the relevant terms to Google for here will help you in making the decisions in homework to identify the localizations of each CA protein in your dataset. So a few words about your final project. You may have noticed that you have about 25% of small homework and 15% of your portfolio, and then the remaining 60% is the final project. We don't have an exam, but instead you are required to make a fairly large project which would end up with the result of 10 to 15 pages and which would take you several days putting into practice all the methods you have learned thus far in the course. And the deadline probably would be in late November or early December. And the 60% of your final course score would be given as 60 points in the project grading, and about 15 points would be for describing and discussing the methods, writing a little bit about how they work, and if there are many options, why did you use a specific one for, let's say, multiple sequence alignment. And then half of your points come from actually completing the analysis, making the figures and presenting the results, and the remaining quarter is for talking about the results, interpreting them, and also reflecting your learning, what you learned from specific parts of this project. The idea is that all of your work would be focused on a single protein and a single gene, and you would be doing many different analyses with the same target. And I'm recommending a human protein or maybe a dog protein or mouse protein that has a known medically relevant phenotype, maybe a disease association. And if there are multiple genes in a family, you might look at several of them at once, but anyway, one protein is certainly enough. And we have a number of tasks which are intended for everyone. This is the list of obligatory tasks starting from here, listed as from 1 to 6. Oops, sorry, 1 to 8 actually. And then there's one elective task. So out of the options of A to D, you are required to choose just one, which would be your elective task, which we could call like task 9. But anyway, the things you are supposed to be doing here are pretty much everything we have gone through in the previous weeks in the course. So you need to use databases to find a suitable gene with the disease association and retrieve the protein sequence from UniProt, I would guess. And because your protein is going to be unique, you need to stake your territory and announce which protein you will be working on. So everybody will be creating unique work, and it's okay that you study together and learn the methods and figure out how to get the results. But because you have your own targets, so you need to repeat the work with a different protein or a different gene, and then get unique results for your own report. So this is pretty nice. People feel that putting all their skills together for the purpose of studying really thoroughly one single protein really improves the learning of the things you've tried very briefly throughout the course. And obviously human is the most studied model organism, so it would be easiest to find information on a human gene. But I don't blame you if you choose a dog protein and then find the disease associations and functions by extension looking at the human genes. But obviously you definitely would need to find the dog disease first and find the associated proteins and choose one of them. If you use human, so there is online Mendelian inheritance in men, which is a lovely resource for all kinds of phenotypes. And then in UniProt, there is a section called Pathology and Biotech, which you might have used in the UniProt exercise already. And so there's a requirement for an introduction of your protein and the disease, and I'm asking for a molecular weight and isoelectric point of your protein, which the isoelectric point is related to the protein being acidic or neutral or basic. And there is a tool which I'm naming here, which helps you get them. Molecular weight you might get directly from UniProt, but then using the calculator shows you that it can be done even if it's an unknown sequence which is not in the database. And for your discussion, it would be good to figure out what are the reasons why the true molecular weight might not be actually the same as the molecular weight calculated based on the amino acid sequence alone. In case of multiple isoforms of a protein, you might want to figure out how to decide which is the canonical version. Again, if it's a well-known human protein, UniProt is probably presenting the canonical version in the database entry and then giving the alternative versions as footnotes in the same sequence entry. Okay, InterPro search and database annotations are one way of figuring out the conserved domains you have there. And you might find some shorter motifs using InterPro, so feel free to discuss them at any length in the same way we looked at the example protein in the InterPro search demonstration. Okay, then you need to find the relatives of your protein. You can do a blast search within the RefSeq only. Otherwise, using the whole NR is going to give you a lot of stuff. And we have some instructions of how to filter this, but I'm not going to discuss everything here. And then, even if you do the filtering of your hit list so that you don't have proteins which are 99% identical to your query sequence, so there might be still other proteins which are very closely similar to each other in your list of hits. And to make a kind of a representative selection of proteins in a protein family, it's a good idea to use some tools for filtering for high identity. And I'm giving you here two options for doing such filtering, and this is something that we didn't discuss during the course, but it will be something new to try during the project. And in order to make a nice compact alignment, I'm asking you to play around with the identity threshold so that you have just 10 to 15 sequences left after you have used filtering for fragment hits in BLAST. Okay, and multiple sequence alignment is one of the key skills in this course, so you are definitely required to make one once you have collected your sequences. Don't take your first MSA. It might be that you still have something that is not of full length, so you might first do the alignment, then decide to take out something, and only then make the final MSA for your work. And in the report, you probably want to include a screenshot of your PDF, which has been created with CView, but you might also want to include the actual alignment PDF, which is of a better quality, as another file in the report, or an attachment for the report. And even a cluster formatted MSA coming directly from the EBI servers, or copy pasted from a text editor is okay. And finally, the last of the obligatory tasks would be making a simple phylogenetic tree to show that you can use Mega or IQ tree, or even Mr. Bayes if you choose to learn it. And this is probably one of the technically harder parts, so you might find yourself asking questions in the forum once you are here. But again, working together might be a good idea if you want to figure out how these things work, if you didn't already try them in the section of phylogenetics. And then in the elective task, so we have a kind of a trick questions here in A. So if you know basic biology, what is wrong in translating a protein into a nucleotide sequence?
It's not called translation, it's reverse.
Yeah, that and then what do you think of the certainty of doing the reverse translation as opposed to translation?
It's about the amino acids. They are like there's it's about the codes. I don't know how to express myself. Yeah, there's fewer amino acids than the codes.
Exactly. So each codon is specific for one amino acid, but there are multiple codons for many amino acids. So you don't know which one of them is going to be or which one has been in the DNA sequence. So the only way to do the correct presentation of a nucleotide version is to find the database entry for the original coding sequence. But what we do for the elective task A, so it is I should rewrite this not into a translation, but transforming a protein MSA into a nucleotide MSA. So you need to retrieve the coding sequences corresponding to your protein sequences, and then there's a software called PAL2NAL, protein alignment to nucleotide alignment, which is using your alignment made at the protein level, and then it's substituting each amino acid with the codons it finds in the in the coding sequences you provided as the second file for this program, and you come up with a nucleotide alignment in that way. And the reason for this exercise is that nucleotide alignments are really uncertain. They have a 25% chance of having a random hit out of the 4 nucleotides. And also, it's biologically not feasible to have gaps which are 1 or 2 nucleotides, but 3 works because it's one entire code and it doesn't mess up with the reading frames. And likewise, 6 nucleotides is a reasonable gap corresponding to a gap of 2 amino acids. And nucleotide alignment programs are usually not aware of this limitation. I heard there are some which are kind of trying to emulate the codon behavior of the sequence and do the translation on the fly and mind the gap length. But in practice it's easier and the programs work faster, and the protein alignment programs are better algorithms directly. So do your protein alignment with Clustal Omega or whatever you decide to use, and then using Paltunal to create the nucleotide alignment. And the reason for making a DNA alignment would be that maybe your sequences are highly similar, so that you have more than 90% identity between all of them, and that means that your alignment comes up with a very small amount of informative columns that can be used for the phylogenetic algorithm. But if you do the nucleotide alignment, it will also contain synonymous substitutions or silent mutations, as they are also called, changes usually in the 3rd nucleotide position in the codon, which lead to the translation into the same amino acid but still a difference in the nucleotide sequence, and that gives you a chance for a higher resolution phylogenetic tree in the case of highly similar sequences. But this is not what you don't need this for the approach we have here. You are trying to get a protein family which has quite a bit of variation between different species, so that you get the protein level phylogenetics working very nicely. Okay, then an analysis of sequence diversity, once you have done your decrease redundancy in the, where was it, step 6 maybe? Yes, it was the step 6. So you can get a percentage identity matrix from decreased redundancy, and you also get a percentage identity matrix from your alignment programs if you use the tools at EBI. One of the possible output formats is the PIM. And if you take the percentage identity matrix into Excel and use conditional formatting, you get a fairly nice colored view, which is able to show you some things of your protein family. So for instance, here we have a list, a group of fairly similar sequences, all with green sage for the mutual similarity. Then we have 2 sequences here which are quite far from anything, so that they are mainly red. And then we seem to have a group of 4 sequences here which are in the shades of yellow, and then there are other 3 which are not very clearly related to each other, but they are not closely related to the group of 4 and not closely related to the group of 5 here. So maybe you'll find something similar in the case of your sequence set in this final project. And this might be a little bit tricky. You need to in case of Finnish locality, because we use decimal comma instead of decimal point, so I have to do a final find and replace for my file to change the decimal points to decimal commas, and only after that Excel will be able to use that. And also because comma is the separator between data items in the American and UK localities, but in Finland and maybe in Germany, don't know about Hungary. So the comma is reserved as the decimal separator, and therefore the comma delimited list is in fact a semicolon delimited list. So first you have to replace the commas with semicolons to get the separators right, and after that you need to change your periods into commas to get your decimal numbers right. And after that you are good to go with this conditional formatting. Okay, if you decide to use Beast or Mr. Bayes to make Bayesian phylogenetics, that's another option for you for the elective tasks. And then finally, studying the structurally known or knowable regions of your focus protein, that's one thing. So one idea here might be to use a blast search of your protein sequence against the sequences in PDB, which is one option in the NCBI BLAST, or maybe you might look at the AlphaFold models. This is something we discuss in much more detail in Introduction to Structural Bioinformatics. If you find an AlphaFold link in UniProt, that might help you in figuring out the structures and presenting some of those structural images and discussing the models, figuring out which parts in your sequence, maybe not all, are easily modelable. Okay, and then there are some ideas of where to use. Most importantly, do not use links which are for search results, because they usually expire in a day or two, and at the longest they remain for a week. So please do not include the links, but instead you need to copy your results and include them as tables or figures in your report. And in case of alignments, you may have noted that an aligned sequence in MS Word is a difficult thing, so it will lose the columns. You need to choose a specific font like Courier or Courier New to have a fixed width for each letter, and then your alignment looks aligned. Or as the other option, make a screen capture and just include an image of the alignment. Okay, this is more or less everything I want to say of the final project. Now, once you read through the instructions with more time, so you might realize that there's something more you need to ask, so we can talk about them in our final meeting, or you can ask the questions in our forum.